So thank you, Hugues, for the invitation. So it's always a pleasure to be part of this journey. So I'm going to present you recent work that we did in lab, dealing with E. coli bacteria that are associated with Crohn's disease, and I will illustrate you in few vignettes of the life they live within cells. Immune cells, actually. As an introduction, I should mention that these bacteria were identified in patients with inflammatory bowel disease. This is chronic inflammation of the digestive tract. Among them, you have Crohn's disease and ulcerative colitis. And the cause of this disease are not completely understood. You have immune and genetic determinant, environmental reason, and today we will focus on the microbiological link with this disease. So this microbiological link is a dysbiosis. So you should know that our digestive tract is rich of microbes. At the equilibrium, these microbes are beneficial. They have anti-inflammatory properties. And in patients with Crohn's disease, there is a dysbiosis, and you lose most of the good bacteria. And these patients, they get an increase of pro-inflammatory bacteria, pathogens, or commensals. A reduction in the protective mucosal barrier, activation of immune response. And 25 years ago, the group of Arlette Darfay-Michaud in Clermont-Ferrand identified a bacteria, an E. coli, that is called LF82, in this patient. And this bacteria became the reference strain of this group called adherent invasive E. coli. And this is this group of bacteria we are interested in. This IEC, adherent invasive E. coli, are not defined by their genotype. I will come back to that, but they are only defined by these three types of properties. So they adhere much better than other E. coli to epithelialized cells. They can invade them, and they survive and eventually multiply within macrophages. In this environment, they induce the production of tons of TNF-alpha, which is a hallmark of the disease. And among other things, this is why this step is interesting clinicians. So in the lab, we have a small collection of IEC bacteria that were collected from patients. So they survive all these tests. And Emma, who's a PhD student in the lab, did an analysis of the genomic content of this bacteria. And on this small cartoon, you see that they belong to different phylogroup of E. coli bacteria, meaning that they can be found in any different type of E. coli. So their genome content is very different, and nevertheless they have the same type of property. So this is not a genetic. Being an IEC is not a fully genetic determined. It could be a phenotypic adaptation. This is an illustration of something that some of you that are not working with bacteria may don't know. Bacteria exchange a lot of genes, and you will see that in the next talk. In this case, you have 15 strains here, and in total they have 9,000 genes, and just 30% of them are present in each strain. 30% of them are present in only one of these strains. So this is to illustrate the diversity. So where this IEC come from? Apparently, they are commensal bacteria that just benefit from the disease to become more pathogen. So they can be found in a healthy individual and animals. So in the lab, we are interested in this survival step and multiplication step in the macrophage. And this movie will illustrate what's happening. So IAC are not very strong pathogens, so usually they don't kill macrophage. They don't detoxify their environment. They just try to survive and make small colony. So the movie is here. And in this macrophage, this is a blue curve here. Oh, no, this one. The macrophage is not able to limit the growth of IAC, and it will form a colony, while this one, for example, is able to limit the growth. This is the brown curve here. And even after 20 hours, there will still be 4 bacteria. And very frequently, what we also observe is a killing of the bacteria, and the macrophage wins. So what is happening when the bacteria are able to grow and form a community in this macrophage? So this is this type of community. You have a macrophage here with two big community in this case. They are contained in phagolysosome. So most of the time, and we demonstrate that at the population level, but also at the single cell level, these environments are acidic, oxidative, they lack nutrients, and they are full of toxic compounds. Nevertheless, the bacteria are able to form community, and you can appreciate on this image that these communities are not homogeneous. This is a biosensor that we use to reveal the acidity of the community. So if the bacteria feel acidity, they turn green, but it's a short time half-life of GFP, so you can get red bacteria if they are not anymore in acidity. So these vacuoles, this community has encountered some acidity because some of the bacteria remain green, but most of them, they turn red, so now they are not in acidic compartment. While this one, all the bacteria are in acidic compartment. So we worked a lot on this community of the form, and just to summarize that, on this slide, this is just to mention that they correspond to multiplication of the bacteria. It's not just aggregation of small vacuoles that aggregate to form big vacuoles. We can detect a sign of multiplication in these vacuoles, such as division rings that are formed in this bacteria. So, what are these communities? This is the main topic of my talk today, and I will illustrate that they are quite robust. So on this first, this is a FRAP experiment where we photobleach this part of the community and look at the recovery. And you see that you don't recover it, suggesting that this is quite robust and bacteria are not free to move in this vacuole. And another example of this robustness is what you can see here. This macrophage will die. This is what you see here with 2 bacterial community, and they survive the death of the macrophage. Even if there is antibiotic in this medium, and they are captured by other macrophage, and then will be mixed with the community that is present here. So this thing seems to be quite robust, and this brought us to consider that there might be biofilm community. So I don't need to introduce what a biofilm is. I just need to introduce what this complex cartoon is the full biofilm formation pathway in E. coli. And basically, you can divide it in 2. Here, it's the gene involved in motility. Here, it's the gene involved in matrix production of the biofilm. In green, you see that all these parts turn green, suggesting that the bacteria in the macrophage, so this is RNA-seq data, the bacteria in the macrophage, they produce matrices while they repress motility. And what you have here is in red is a regulator of this cascade. And what we identified with TNC approach, something I'll come back later, but something that allows you to identify essential gene for specific growth in a specific environment. We observe that this regulator of the biofilm cascade are very important for survival in the macrophage. So biofilm are also formed with a matrix, and we try to identify this matrix within these small vacuoles with different type of labeling. And this is a few examples. We have a labeling of the extrapolysaccharide matrix with WG lectin, and you can see this very nice labeling on each bacteria inside this vacuole. We can also detect curly fiber that are other type of component of the matrix around IEC bacteria, but not around laboratory strain bacteria that have been in the same macrophage. And we demonstrated that altering the biofilm pathway by deleting one of the regulators indeed reduce survival of this bacteria in human-derived macrophage from monocyte. So what we think is that this community called intracellular communities are beneficial for long-term survival of the bacteria within macrophage, and eventually to get them protected from the environment, from other even cells, from phages, from the flush in the gut, and etc. And this is what Emma observed, that we can get IBC for more than 96 hours in this macrophage that stay for 96 hours. They are not that big, they can contain up to 100 bacteria, but in average they contain 30 bacteria, each of them. And this brought us to consider that this vacuole, these communities, could be a bit like an ecosystem for this bacteria. And what can they do in this ecosystem? And we start to investigate this aspect. And this ecosystem could be form of one single type of IEC, but also from different IEC, because it is known that patients infected with patients presenting Crohn's disease and infected with IEC can present up to 3 different IEC in their feces. IEC of different phenotype, that's what I illustrated before with the acidity, and also bacteriophages, because these bacteria are producing phages from prophages. So I start with this story about phages, something that we did in collaboration with the group of Marie-Agnès Petit at the INRAE in Julian-Josas. Actually, what they get interested in the prophages present in the genome of LF82, the reference strain of the IEC. They observe that 5 prophages are present there, and these prophages are active. They can produce phages, and among them, one of them is very active. So in LB culture, you produce like 10 to the 8 phages per ml of culture. This is quite high for phage. And what they observe is that you can even induce more this phage with treatment with ciprofloxacin that will damage DNA and induce the DNA damage response in E. coli called SOS response. And what they have been able to show is that if you look at the growth curve of these bacteria, if you treat them with cipro at a dose where cipro is not really killing the bacteria by itself, the bacteria are dying, and this death is led by induction of the Gali prophage, since when you delete this phage, you restore growth. Not fully, but you restore growth. They came to us and say, okay, these bacteria, they produce ton of phages in the macrophage, in the vacuole, you should have oxidative stress and blah, 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 and this should lead to death of the bacteria, etc. So we checked actually if the condition for phage induction were present in the macrophage, and on these two slides, I will illustrate this result. Indeed, the conditions are favorable for this prophage induction. All these genes that are involved in DNA repair are essential for the survival of the bacteria in the macrophage. So you need DNA repair in this condition to form the community, and you induce all the genes from the SOS response regulum. So this is RNA-seq data now. Suggesting that, yes, the conditions are met to induce the prophage. So the surprise was this result. We compared the viability of the bacteria inside the macrophage when they can produce this phage or when they do not produce this phage. And there is absolutely no difference in between these two conditions, suggesting that even if the bacteria are able to produce the phage, either the lysis is not happening or something else is happening. And actually, we were able to prove that the phage is actually not produced, and this is linked to a particular regulation of the whole phage operons. So this is all the gene of the phage. This is a RNA-seq data set. And I didn't put it here, but in cyprofloxacin, all these genes will be overexpressed. And what you see that in the macrophage, only the first part of the phage genes are produced, and these ones are repressed. And therefore, some genes involved in the early step of phage production are produced, but not the last stage. So in the macrophage, you don't produce viral particles. And we are working on that to try to understand the regression for that with Marine's group. So that's what I just told you. We have a repression of phage production in this particular environment. And then we start to consider what's happening when we mix different bacteria. And the first observation that we did was this one. We mixed green and red bacteria, a little bit like Alexandre presented before. And we infect macrophage. You see that macrophage at 1 hour post-infection contain green and red bacteria. And when you look at communities, most of the time, these communities are segmented with red sector and green sector, suggesting that they don't come together, even if the vacuole fuse and etc. And we were able to imply the biofilm matrix into the structure. But more interestingly, we start to play with nanoparticles to try to characterize the biophysics of this environment. And we obtain very, very, very surprising results. So this is small nanoparticles that fluoresce in red when they are in acidic compartment. And this is bacteria. And for example, you see this vacuole that is completely split in two, suggesting that the diffusion of the red nanoparticle is not allowed in the region occupied by the bacteria. We have different configuration, but you can see that also in this thing. So it's very common. We have configuration where the bacteria are in the middle and the particles are around, and we are investigating using this type of tool to investigate the diffusion of molecules, and it could be nutrients, signaling molecules, CD4. We are a lot interested in diffusion of CD4, so elements that allow capture of iron that are produced by the bacteria that can go in the vacuole, but also in the cell to modify the signaling of the macrophage. What we also observed when we mixed bacteria that came from different phylogroups, for example, from the B2 phylogroup or from the A phylogroup, is that they form community with different characteristics. I don't go into the detail here, but for example, this one is not able to form a matrix that is labeled by WGA, while the red one is able to form a matrix that is labeled with WGA in purple. So you should compare the interior of this vacuole here that is occupied by green bacteria, but without matrix compared to this vacuole here. And what we observed is that most co-infection experiments lead to asymmetric vacuoles. So either vacuoles that are fully green with no red, or fully red with no green, or with a lot of red but very few green bacteria. And this is a lot above random distribution of the bacteria in this thing. We are investigating this aspect with different type of experiment, and this is a very recent experiment that we did. We infect with two type of bacteria and follow the dynamics of the community over time. So this is plenty of different communities that are followed, and you see here the amount of fluorescence allows you to determine the number of bacteria in a community, in a given community. Most of them, they stay homogeneous in this period of time, between 1 day and 2 days post-infection. This is true this is true for both bacteria. But when you look at mixed community, you see very different behavior. So in this case, this is a community formed by phylogroup A bacteria, and you look at the number of B2 bacteria in that, and you see that it stays low. Eventually, they disappear from this community, while this is the opposite in the other context. So it's an IBC from B2 bacteria, and you look at the behavior of A bacteria, and they start by being low, and they get control of the community after a while. So you have an ecological dynamics that is starting in this community. And we are trying to investigate that, and a way to investigate that outside of the macrophage was to build microfluidic chambers where we can generate microbiofilm at the size approximately of this community we observe in macrophage. So this is the work of Stefan in the lab. He built, with a bit of difficulty, a very small chamber, like less than 100 micrometer large. This is a channel that brings the bacteria, and you can seal this channel with oil, for example, and allow the bacteria to multiply within this fix, or you can flush any kind of things you want in these channels, for example, antibiotics, to see if the community that the bacteria form will tolerate antibiotics. This is what we observed. This is one of these chambers. And indeed, we observe the formation of aggregate of bacteria that may resemble biofilm. So yet we are not fully able to say if they contain matrix or if it's just aggregation. But nevertheless, some of these bacteria, they form communities with aggregative bacteria. Some form a different type of community with chains of bacteria, and some remain mobile in this chamber. So if I show you the 3D representation, so we have this kind of thing, but it's more impressive on this one, where you can really follow the chains that this bacteria is making. And in good correlation with what we observed in the macrophage, we can create sectors. So differently from what Alexandre presented, in our case we have no flux. So these 6 sectors are emanating without flux, and they they divide the different type of bacteria. So we can reproduce in vitro what we observe in the macrophage, and we are now trying to use this tool to decipher the mechanics behind this community evolution, formation, evolution, etc. And I will finish with that. We can also come back to the fate of the macrophage of the different bacteria according to the type of community they form, either it's a community structured by biofilm matrix or not. So this is the movie I already showed you before. This is the only fate that we observe for LF82, the reference strain. It very rarely kills the macrophage and do not disseminate in the exterior medium, but can be captured and transferred from macrophage to macrophage. This is a different IAC, and this one doesn't form a matrix. And what you see is that the behavior is a bit different. Now, this one can disseminate within the cytoplasm of the cell. This is what you see here, and colonize the whole macrophage before killing it. And I finish with this group. Sorry. And this group form a biofilm type matrix, but is able to disseminate by killing and exploding this matrix. This is what you're going to see here. Yeah, the macrophage is going to die here. You form big bubbles, and the bacteria, they disseminate 1 and 2, etc. So this is really stereotypic. I mean, these bacteria always do things like that. This one always does things like that, and this one always does things like that. And it changed a lot the outcome of the infection when we look at macrophage in ex vivo, in vitro assays. And we think, and our collaborator in Clermont-Ferrand, they think that this could also be instrumental to understand the difference in inflammation in patients. And they are trying to address that in tissues from patients. Well, I stop here. I would like to thank my team and my collaborators. So all the phage story was performed with the group of Marianne Petit. All the work with the microbiofilm, all the work with the microbiofilm, the microfluidic system, is performed in collaboration with a group of Sylvia, with whom we will try to do some mathematical modeling of what's happening. We had a lot of help from the imaging facility. And in the lab, Gaël and Victoria started this project. And now Emma, Osni, Kevin, Nicole, Stéphane, Sylvie are involved in this, and the other one, they are working on chromosome biology. Thank you very much.